We're going to travel back in time to 1928, to a scientific mystery that honestly turned our understanding of life completely on its head. And it all kicks off with one very, very weird question. Yeah, I know what you're thinking. Sounds like a headline from some old detective novel, right? And in a way, it kind of is. This was the puzzle a scientist named Frederick Griffith was staring down. And believe it or not, the answer he found is the reason we can do incredible things today, like genetic engineering. So, come on, let's meet the suspects. The whole case centers on this one little bacterium, Streptococcus pneumoniae. Now, you can probably guess from the name, this is the bug that can cause pneumonia. But Griffith noticed something really interesting. It wasn't just one type of this bacteria. There were two totally different versions. You could think of them as our main suspects. All right, let's get our suspects straight. First up, you've got the S strain, and the S stands for smooth. And this thing is bad news. It's got this sort of slimy, protective coat that makes it a real killer. Then you have the R strain, R for rough. No coat, totally harmless, kind of the innocent bystander in all of this. So Griffith, acting like a proper detective, decides to do some basic legwork first. He just wants to establish the facts, right? To see what these two strains do on their own, he used mice for his tests. Okay, so here are the first clues. Clue number 1, he injects a mouse with the live, deadly S strain. And, yep, just as we thought, the mouse dies. No surprise there. Clue number 2, he injects another mouse with the harmless R strain. The mouse is perfectly fine. It lives. Okay, still makes sense. Clue number 3, he takes the killer S strain, but this time he cooks it, kills it with heat, and then injects it. And guess what? The mouse lives. So the rule seems pretty straightforward, right? A dead killer is no longer a killer. Everything is lining up exactly as you'd expect. But this is where the story takes a sharp turn. This is where things get weird. Griffiths decides to mix things up, to try one final experiment with a combination that, logically, should have been totally harmless. I mean, seriously, what happens when you mix two things that are harmless? You've got the dead S strain, can't hurt anything, and you've got the live R strain, also can't hurt anything. So you put them together, and you should get nothing, right? Just a harmless cocktail. At least that's what everyone, including Griffith, expected. So that's the final test. He takes the dead S strain, harmless, mixes it with the live R strain, also harmless, and he injects that mixture into a healthy mouse. And the mouse died. Just died. This was a total shock, a complete plot twist. How could two harmless things create a killer? But it got even crazier. When Griffith looked at the dead mouse's blood, he found live killer S strain bacteria. The harmless R strain had somehow become the killer. This absolutely baffling result meant something profound was going on. Griffith realized he had accidentally discovered a fundamental secret of biology. He gave it a name, the transforming principle. His theory was this: something, some actual physical substance, must have passed from the dead killer bacteria to the live harmless ones. And this mystery substance, whatever it was, it carried the instructions, the blueprint for how to become deadly. It literally transformed the good guys into bad guys. Okay, here's a simple way to picture it. Imagine the dead S strain bacteria are like a bunch of discarded cookbooks lying around. The books themselves can't hurt you, right? They're just paper. But inside, there's a recipe for that deadly smooth coat. Now, along comes our harmless R strain bacteria. They're like chefs without a recipe. They find these cookbooks, they read the recipe, and they start to follow the instructions. And voila, they cook up that deadly coat and transform themselves into the killer S strain. And that's what killed the mouse. So why is this so important? What's the bottom line? Griffith proved for the first time ever that a physical recipe, what we now call genetic information, could be passed between organisms. This is huge. This process is happening all the time. It's how bacteria share traits, like becoming resistant to our antibiotics. And on the flip side, it's the exact same principle we use in labs today for genetic engineering. He basically uncovered a secret language of life. So Griffith solved the first mystery. He proved that information could be transferred. He opened the door to a whole new world. But in doing so, he created an even bigger mystery for the next scientific detective. What was this transforming principle? What was this recipe book actually made of? Well, that is a story for another time.